Welcome to House of Sweden and to this live webinar, A Swedish-American Journey of Innovation in Cancer Research. My name is Karin Olofsdotter and I'm the Swedish ambassador to the US, and it is my pleasure to invite you all virtually to the House of Sweden. Of course, I wish you would have been here live, but this is the best we can do given the times. Today's seminar will focus on the life science sector. I think never in our lifetime has there been such a high global interest and demand in the life science sector. How vaccines and medicines are developed and tested, the ethical aspects, the best treatments, etc. We're all following these topics in the media and of course learning new things every day. Health and life science is a prioritized sector for the Swedish government. We are a major player in the life science arena with a thriving life science environment. A fundamental part of success for the life science sector in Sweden is the collaboration between academia, government, and industry, the so-called triple helix model. This has benefited our economic prosperity and also advanced our healthcare system. Sweden is moving the boundaries of life science forward. Over the years, we have created distinct expertise in health and digitalization, and we have collaborated with scientists worldwide. This collaboration has resulted in world-leading life science companies such as Getinge, AstraZeneca, Elekta, and many more. Getinge and AstraZeneca have both been part of the work to fight the pandemic, not least here in the United States. I really believe that the life science sector is a good example of where international collaboration is essential. Sweden and the US are a perfect match in this regard. We are both innovative countries with world-leading companies and universities. We have already seen many important ongoing joint projects. One example is the US Cancer Moonshot Initiative that Sweden is part of, which was signed by President Joe Biden during his time as Vice President in the Obama administration. We really look forward to continuing to develop this project together and also, of course, further explore areas of potential research cooperation between our two countries. So today we will discuss the following topics. First, we will start out with the Swedish innovation model with speakers from the industry, academia, and government. Secondly, the cancer sector with representatives from two Swedish cancer treatment companies, AstraZeneca and Oncopeptides, and with an extra focus on one form of cancer, myeloma. Thirdly, and lastly, we will discuss the Swedish-American collaboration through Dana-Farber and Harvard. I really look forward to interesting presentations and discussions, and all the work you do is very important for all of us. Thank you so much.
Varmt välkommen. Warm welcome. You're attending the Swedish-American Journey of Innovation in Cancer Research program. My name is Ola Landgren, and I'm professor of medicine at the Sylvester Comprehensive Cancer Center at the University of Miami in Miami, Florida. Today's program is a hybrid meeting live from the House of Sweden, the Swedish Embassy in snowy Washington, D.C., live from here in sunny Miami, and virtual across the rest of the United States, Sweden, and beyond. Together today, we will shed light on how collaborations between industry, academia, and society in Sweden and the US can serve as a powerhouse to drive innovation and science from the laboratory to the bedside. Before I opened the program here today, I was asked to give a very brief background about myself. Jag är född och uppvuxen utanför Lund. I was born and raised outside the city of Lund in southern Sweden. After completing my MD and PhD at the Karolinska Institute in Stockholm, Sweden, in 2004, I came to the National Cancer Institute at the National Institutes of Health in Washington, D.C., with a plan to stay for a 2-year postdoc program. After completing my postdoc, I was recruited to establish a world-class myeloma program with focus on early drug development. And in 2014, I accepted a position at Memorial Sloan Kettering Cancer Center in New York City to create and expand a myeloma program with focus on drug development and studies on biology. Last year, in 2020, I was recruited by Sylvester Comprehensive Cancer Center at University of Miami as chief of their myeloma program and leader of the experimental therapeutics program. While I have lived and worked in the United States for over 17 years, I have maintained close collaborations with many colleagues and friends back in Sweden. And based on my experience, collaboration between industry, academia, and society in Sweden and the US can serve as a powerhouse to drive innovation and science from the laboratory to the bedside. For those of you who are not familiar with multiple myeloma, it's the second most common hematologic malignancy, which affects about 130,000 people annually worldwide. And currently, unfortunately, we lack an established curative treatment. During today's program, multiple myeloma will be used as an example to visualize how research collaborations can transform patients' lives. Today, we have an exciting program which gives you an overview of the unmet clinical needs for patients with myeloma, important patient perspectives, and 2 panel discussions with representatives from industry, academia, and society. This program will be recorded and shared online, and you are welcome to post questions via Zoom, and we will try to answer as many as we can today. Any remaining questions will be answered after today's program. And I ask all the participants to kindly stick to the allotted time. Please do not go over. And I will do my very best as a moderator to ensure we stay on time. Now, I would like to introduce to you my Cancer Institute in Boston, Massachusetts. Paul will give a 10-minute keynote speech on multiple myeloma. Paul, warm welcome.
Great. Thank you so much, Ola. It's an absolute pleasure to be with you. Thank you for that very kind introduction. To join you, Ola, as well as, of course, Joachim, Professor Henrik Johnson, and of course also my mentor and colleague, Dr. Kenneth Anderson. With that in mind, I wanted to emphasize today in the 10 or 15 minutes that I have allotted how in multiple myeloma we've seen a transformative journey in the last 2 decades that has truly impacted on our patients' lives in a very, very favorable way. Next slide, please. David, if you could advance the slide, David, please. Thank you so much. These are my relevant disclosures. Next slide, please, David. Thank you so much. So just for those who may be less familiar, I just quickly summarize on this slide the nature of what multiple myeloma is. As Ola mentioned, it's the second most common hematologic cancer after non-Hodgkin's lymphoma, and it's characterized by an uncontrolled neoplastic proliferation of plasma cells. And this is particularly important in the context of the bone marrow microenvironment. These are derived obviously from B cells and in my view constitute a queen of immunologic cancer in the sense that they are terminally differentiated and an absolutely centripetal part of our immune system. Very importantly, malignant plasma cells secrete abnormal antibodies or a so-called monoclonal protein or M protein, and this is associated with profound immune dysfunction, organ dysfunction, and of course within the microenvironment of the bone itself and the bone marrow, there's profound disruption of cortical bone and components of blood formation. This in turn results in tremendous morbidity and very high mortality in the absence of effective therapy. Next slide, please. So to summarize that, this is in a quick summary of the impact of myeloma. It's truly a systemic disease with an impact through the bloodstream in terms of infection and coagulopathy. Very importantly, it attacks the kidney. And of course, bone disease is a profoundly morbid manifestation of this illness, where we see our patients experience significant pain from disruption and damage to their cortical bone. And in fact, myeloma is defined in its active form by what we call CRAB features, characterized by renal impairment, anemia, bone disease, and hypercalcemia. So in its uncontrolled fashion, it's an extremely dangerous and incurable disease. Next slide, please. Now, in terms of risk factors, it's important to recognize that this is, as Ola so nicely pointed out, or importantly pointed out, this is a much more common hematologic cancer than is perhaps appreciated. And sadly and tragically, there still remains a mortality of around 13,000 deaths in the United States every year. And there are at least 32,000 new cases estimated for last year. And unfortunately, what we've seen is a dramatic improvement in survival rates over the last 20 years that we'll spend a little bit more time on. In the context of the global incidence of this disease, it's at least 140,000 cases, although this is probably an underestimate. In terms of median age at diagnosis, it's 69 years, And an important aspect of risk factors not only includes increasing age, but very importantly, this is truly a disease of ancient peoples. And in that regard, African American ethnicity is very important and an important risk factor for this illness. Obesity is also another factor that's involved, as is family history, as we're increasingly realizing, and underlying conditions such as monoclonal gammopathy of uncertain significance, a much more prevalent disease, is an important risk factor for myeloma. Next slide, please. Now, in terms of understanding the biology of this disease, it's important to recognize that it's classically described as multiple myeloma. It is not one disease, but many. It's highly complex at diagnosis and at relapse due to genomic events and what we call clonal evolution. And I seek to summarize this in this particular slide. And what this illustrates is the tremendous mutational thrust of this disease. On the right, you can see what we call whole genome sequencing, which in a newly diagnosed patient constitutes around 5,000 mutations in their myeloma. And then after treatment and at relapse, in this particular patient, that mutational burden dramatically increases to 12,000 mutations. So this provides a substantially challenging problem for us as caregivers and as hematologists to treat this very challenging process. However, the good news is that with risk stratification, the recognition of clonal heterogeneity, and the individualization of treatment with the advent of novel therapies over the last 3 decades, in fact, and in particular the last 20, real progress has been made. Next slide, please. So as we think about challenging this disease, I want to especially emphasize the role of the microenvironment in this disease, understanding that it's not just the tumor cell, but also the bone marrow microenvironment that's a critical target for our therapies, has really, in my opinion, led to a revolution in novel treatments. And I especially want to acknowledge Professor Ken Anderson, my mentor and partner, who has obviously been central in the advances of research in this regard. And in that respect, too, obviously, a number of other groups over the last 2 decades have focused on this critical aspect of myeloma pathobiology that has opened the door to advances in treatment. Next slide, please. Now, as we think about this in the context of treatment, I use this particular pictorial, just again, the complexities I don't want to go into in the interest of time, but simply to emphasize to the audience that by targeting various aspects of biology, we've been able in a combinatorial sense to, in a multimodality fashion, improve the therapeutic index of our combination therapies and dramatically improve outcome. And I especially want to acknowledge my colleague, Dr. Giada Bianchi, who is the first author of this particular paper, in which we sought to summarize this effect. And what you can see from arm A, as it were, at the very core of this pathobiology slide, is the targeting of the proteasome. Then as you move clockwise around the cell, you can see that there are a variety of other strategies that involve targeting not only the immune system, but also key aspects of the pathobiology of myeloma that we've been successfully able to exploit. But critically also around that has been the impact on the microenvironment. But very importantly, targeting the stemness of myeloma as a functional unit has become also an important focus of our research. Next slide, please. So another concept to share with you is that as we treat multiple myeloma, this disease is not a sprint, it's a true marathon. And there's now the ability to offer a longitudinal approach to the management of patients to improve outcome. And as we think about our treatment paradigm, we've evolved from initial treatment and transplant as a mainstay of therapy for the younger patient, with unfortunately after that treatment failure leading rapidly to death, to a much different paradigm where we now have a much more longitudinal view of disease management, where multiple therapies allow us to prolong survival in a profoundly meaningful way for our patients. But this also beholds us to think strategically about how we manage the disease. And whilst cure remains elusive, there's an increasing proportion of patients who enjoy what I consider almost a functional cure, where with continuous therapy, and again, this highly rational novel approach to the biology of their disease, long-term survival is possible. However, there is unfortunately a very high-risk group who remain a fundamental challenge in terms of therapeutic outcome. Next slide, please. So as we think about these new therapies, there's a lot to cover in this slide, but in the interest of time, I'll just simply summarize it. In the last 20 years, we've moved into the era of true novel therapies. But to give you some perspective, I seek in this slide to show you that, in fact, over 50 years ago, 60 years ago, was the advent of melphalan, chemotherapeutic that was particularly important as a mainstay of therapy back in those days and transformed management indeed. But we've moved rapidly in the last 2 decades to an entirely different paradigm of biological approaches, including the use of immunomodulators, proteasome inhibition, other next generation immunomodulatory drugs, totally new classes of agents such as selinexor, an exportin 1 inhibitor, and then, very importantly, the advent of the monoclonal antibodies, importantly daratumumab as well as others, and then most recently the excitement around BCMA targeting and immune therapies. And we're going to spend a little bit more time on each of these. And then in the context of our conversation today, just to emphasize the excitement around another new mechanism action characterized by melflufen or melphalan flufenamide as a peptide drug conjugate. Next slide, please. Now, in terms of these agents that have transformed our therapeutic landscape, I want to emphasize the immunomodulatory drugs. I've sought to summarize them here in a simplified form to show how, for example, the transition of thalidomide to lenalidomide has truly led to a transformative impact on survival. Next slide, please. And in that same space, the combination of the immunomodulatory drugs with our proteasome inhibitor platform has equally been profound in its effects on treatment outcome, and in particular with the advent of combination therapies. Next slide, please. And then as we think beyond this, the monoclonal antibodies have been, in my view, probably one of the most important advances in the last decade, where we've seen the advent of daratumumab, monoclonal antibody that actually was developed by a company called Genmab, based in Copenhagen in Denmark, and it's been our privilege to work with them as this drug moved forward in development, has been truly transformative in the therapeutic landscape. And similarly, other antibodies have followed suit, including the targeting of SLAMF7 with elotuzumab, and most recently with another very exciting antibody, isatuximab, which has allowed us again to take yet another step forward in the treatment of our patients. Next slide, please. Now, with that in mind, beyond the antibodies, exciting new other drugs have emerged. These include panobinostat, a histone deacetylase inhibitor. I've already mentioned selinexor, which had an FDA approval at the end of last year. And then, very excitingly, another antibody also approved last year, belantamab mafodotin, which is a fascinating concept of combining an antibody with a cytotoxic payload and delivering it to the tumor cell. And this drug has provided a means of targeting BCMA off the shelf in a way that's incredibly important, particularly in the context of the current pandemic. Next slide, please. And as we think about next steps and future directions, I do want to emphasize the excitement coming. Venetoclax, a BCL1 inhibitor, entirely novel mechanism of action. We're hoping for approval later this year or sooner perhaps. And of course, very importantly, the CAR T therapies, true cellular platforms of immune therapy that are so promising for those patients who are able to participate in such treatment platforms. And then perhaps even more excitingly are the bispecific antibodies, which again provide this off-the-shelf approach. So really a veritable explosion in new treatment options for our patients, all hopefully improving outcome. And we're very excited by the promise they hold. Next slide, please. And then in the context of today's conversation, it's my privilege to talk to you about yet another exciting, very practical, patient-friendly, and usable approach with a peptide drug conjugate mechanism. This, of course, is melflufen, and I summarize the mechanism action here, this being absolutely innovative technology that we'll talk about more in the to and fro of conversation. But suffice to say that this preclinical development of this drug summarized here, I especially want to acknowledge my colleague Dr. Dharminder Chauhan and our laboratory team led by Dr. Kenneth Anderson, in which we really were able to develop a very exciting preclinical data that really then informed our clinical trial translation that has led, we hope, to the successful development of this drug. But just suffice to say that this provides a mechanism of delivering a cytotoxic warhead to the tumor cell in a highly directed fashion, challenging this stemness that is such an important part of overcoming myeloma resistance, and doing so in a way that's remarkably selective, we hope, and at the same time minimizes side effect profiles for our patients, and at the same time allows us then to really deliver a lethal blow to tumor burden, and at the same time then improve outcome. Next slide, please. I'm just going to very quickly finish on there.
I think okay. This will be the last slide.
Okay, I'll finish there by just thanking you very much to say that's very exciting. I apologize, Ola. I was given 15 minutes on the program, but if that's too short, I fully understand we're running behind time. But I just want to specifically conclude by saying that we're very excited to be here with such an exciting armamentarium of new drugs and to be able to offer our patients improved outcome. My concluding slide was just to acknowledge the vital nature of partnership that has allowed us to do this. So thank you very much, Ola.
Thank you very much for a wonderful talk, Paul. I guess, gave you almost an impossible task to cover this whole field in 10 or 15 minutes. So it was probably on us that we set you up a little bit for a challenge there.
Yeah.
Brilliantly, as always. So I think you had invited a patient also to be part of this discussion here today.
Yes, please. And, absolutely. Curtis, obviously it's lovely to see you. If you could just just for the audience, summarize when you were diagnosed, what your treatment's been, and how your journey's been on melflufen, that would be wonderful.
I will do it very quickly. I was diagnosed with myeloma in 2007, and this was both a kind of a bleak time and a promising time, because at that point, really the only, as I understood it, the only viable option was thalidomide, which certainly worked for me, but produced a great deal of difficulty in many ways. I've always been lucky with myeloma in the sense that the people who do the hard work of creating these drugs always stay a step ahead of it for me. And I've gone through several trials, pomalidomide and most recently melflufen, which worked very well for me and had really very few issues. Obviously, it's a disease that affects people different ways, and so do the treatments. Aside from some fatigue, it seemed to have worked very, very well for 2 years. And I'm very grateful for that and for your help in steering me to it. As it stands right now, we're probably going to move on to something else, and I'm sure we are. And I hope for the same kind of results that we had with this one in the previous trial.
Wonderful. Thanks so much, Curtis. And I think you bring out a beautiful point that it's been otherwise very well tolerated. I'm encouraged to see you haven't lost your hair, which is great. That's a huge advantage of this. I know you and
I'm trying to position my head so you can't see it.
And at the same time, as you say, we're well placed to go on to our next therapy when we need to, right, Curtis? Because we don't have to move on to anything immediately. We'll just have complete therapy and then make a decision about what we do next. And do so in the context of...
I would like to thank you very much for your guidance, and as I said, for all the people in this business who tried so hard in school to learn things. And you did a wonderful job.
It's our privilege, Curtis. Thank you very much. And I think we'll hand back to Ola. Thank you, Ola.
Thank you very much, both of you. This was a lovely discussion, and it's, of course, very, very important to hear the patient's story. That's all it's all about.
We were very convinced that this molecule was something, but there was still a lot of skepticism out there. We have always said, if it's good for the patient, it's good for Oncopeptides.
The only way we can compete with the big pharmas is to be something else.
The soul of Oncopeptides really is in the science and collaboration.
We need to have trust and respect for each other.
We always work as a team.
Science matters, colleagues matter, the patient matters. That's it.
Starting at Oncopeptides really felt like coming home.
Eva was working at AstraZeneca at the time, which of course is a big company.
And a friend of mine said, you should call Jacob, who was the CEO at the time.
She had long industry experience, and while AstraZeneca went through a lot of reorganizations, she would have no problems finding a good position, either within AstraZeneca or at another pharmaceutical company. So why would she choose this small company in a single room with no money?
He was just so passionate about the molecule and the company, so it was very hard not to get caught up in that passion.
We really liked each other. We shared a lot of ideas, and I think we complemented each other really well.
It was instant joy.
I think she wanted an adventure too, and she believed in the molecule.
Imagine taking this drug and helping patients. It felt very real to me.
Fredrik Lehmann, he is and he was the head of CMC. He has been absolutely instrumental in making this happen.
When I started in 2008, we were in a basement in an old building at the Karolinska.
You could only get short-term contracts because they were about to blow the building out, right, and change everything.
4 desks in it and some billy shelves to put our files in.
That were kind of slanting. We had an old server that was basically a scrapped PC that were several years old.
But it was nice. I mean, we were all together in the same room, making our best to get this drug into man. But it was even better when we moved to our new proper office.
A new room that was much, much bigger, but still just one room. But then we shared that big room with 2 other companies.
The only real meeting room was also the coffee place. So if there were an important meeting, nobody could get coffee. And one of the best places for phone calls was actually in the toilet.
We had almost 20 desk spots in that room, and it was approved from a fire safety point of view for 6, I believe. That was the limit. So the landlord was very stressed when they realized how many people we were in that room.
Whenever it is indicated that the gas mask is to be used, the soldier must first hold his breath to avoid leak.
An alkylator is a toxin that attacks DNA in mainly fast-growing cells, like cancer cells. The origin is actually a deadly gas, mustard gas, from World War I. After World War II, doctors realized that they could also use alkylators in the right doses to treat cancer. Joachim's molecule builds on this old knowledge.
The molecule was first synthesized by Joachim Gullbo at Uppsala University.
These molecules are based on very old science, but it also applies more modern knowledge about enzymatic activity in cancer cells.
Joachim is a friend of mine from university, and he would synthesize the compound and then jump on his bike and test it in another lab down the street.
He added something to the alkylator class. He didn't fully know what it was, except that the data was simply much improved.
It has something old, but also something new that makes it trustworthy.
To this molecule, we have connected a little linker, a little piece of a new molecule that makes it go even more specifically into cancer cells.
I think this is an innovative medicine, and it's not only one medicine, it's a platform of molecules.
There are a lot of companies that have more fancier theories, but Oncopeptides will be the one that works in the end, because it's simple, it works.
I don't think I've ever doubted that this molecule actually has something for the patients.
Every single data point we had said, this molecule helps patients.
It just had that thing that you could believe in. I really like the mechanism of action. It makes a difference.
Paul and his team at Dana-Farber, early on they were skeptical because they thought, this is just another alkylator, right?
But as soon as they started testing, things changed.
My collaboration began with Oncopeptides over 10 years ago, and we really had lovely data, both in vitro and in vivo. I started to really appreciate that this drug was quite distinct.
We will start our first of 2 planned panel discussions. And the focus for this first panel discussion is the Swedish innovation model. And this includes the so-called Teachers Exemption rule. And people outside Sweden may or may not know about this. So we will talk about this Teachers Exemption rule, how that has enabled the establishment of several innovative life science companies, including Oncopeptides. So the goal of the panel is to discuss the role of the Swedish model and how it can be leveraged to spur innovation. We have invited 3 participants for this panel, and I will give them each 4 minutes to talk, and then we will have about 15 minutes in the end for discussion. Hopefully the people online will send in questions. I already have a couple of questions, and I will also ask questions. So first of all, I'd like to welcome Dr. Joachim Gullbo from Oncopeptides, Dr. Mats Larhed from Uppsala University, and Dr. Henrik Johnson from House of Sweden, the Swedish Embassy in Washington, D.C. Welcome, all the three of you. And Joachim, the floor is yours.
Thank you very much, Ola. And I will try to summarize my 25 years of experience with Oncopeptides in 4 minutes. To find some inspiration for this short presentation, I dug up my thesis, which was published in 2003. It was a result of a collaboration between Uppsala University, both Faculty of Medicine and Faculty of Pharmaceutics, and Karolinska Institute. And by the end of the 1990s, Professors Hans Ersson and Rolf Levenson at Karolinska Institute had worked with an Italian chemotherapy cocktail, and they were very much interested in this class of compounds. They brought the project to Rolf Larsson and Peter Nygren at Uppsala University, and as a happy coincidence, around that time, I knocked Rolf Larsson's door looking for a project involving medicine, pharmacology, and chemistry. I brought Christina Lukman from Faculty of Pharmacy into the project, and we established a small research group, 5 professors and 1 medical student. And while I did most of the hands-on work in this project, I had 5 supervisors. I didn't have to worry about any funding. They gave me all the resources needed, and they were always there to discuss ideas. We were immediately intrigued by this class of compounds and the effects we could see in vitro. And important to remember here, this was a time period where information technology grew very rapidly. NASDAQ reached records every week. There were a lot of venture capital around, and Hans Wicksell from Karolinska Institute, he realized that this was a unique opportunity for academic projects to be turned into companies and to come to the benefit of more people and to have other benefits than just the academic perspective. He formed a seeding umbrella organization called Karolinska Innovations that should help startup companies from the academia. And that was led by Conny Bugentoft, who happened to be a good friend of Hans Ersson. And when Hans told Conny about this project, he said that we should form a company around it, and Oncopeptides was formed in 2001. We made a series of compounds and saw really good effect. We were intrigued by this and continued to develop. At the same time, Glivec was introduced on the market and approved to treat CML, and everyone for a short period believed that if you could just identify the right target, you could treat and possibly cure every type of cancer. And then the dot-com bubble bursted, and all the money that had been on the table were just gone. It was very difficult to get any funding at all for Oncopeptides. But eventually, Karolinska Development was formed, and Industrifonden joined with a small investment. And for a while, we lived hands to mouth, doing GMP synthesis of the compound and GLP laboratory studies to meet regulatory requirements. And we were able to do a first-in-human study at Uppsala University Hospital and Karolinska Hospital. Despite this, it was quite limited enthusiasm outside our research group. And while we continued with the academic research leading up to a thesis for Malin Wikström in 2007 and Sara Strese in 2015, not much people outside this group paid any attention to the project. And it was not until Rolf Levenson contacted his friends, Professor Kenneth Anderson and Professor Paul Richardson at Dana-Farber Cancer Institute, and Dharminder Chauhan did a dedicated study in myeloma published 2013 that the company really boosted. Around this time, we also had Jacob Lindberg as the CEO, and we were able to attract money from HealthCap and Industrifonden, and we started the first myeloma study.
Thank you very much, Joe. There are so many exciting things to hear about here, but unfortunately, we also have to stick to the schedule, and we started a little bit late. So thank you so much. I will come back with the discussion in a little bit. I would like to hand over to Dr. Mats Larhed from Uppsala University.
Many thanks, Ola. my contribution will not be as exciting as Joachim's. But I would like to thank the organizers for inviting me to this webinar. Many thanks. I have a few comments regarding the role of Swedish universities and the Swedish innovation model. According to the Higher Education Act, the universities' assignments in Sweden include the tasks of collaborating with the surrounding society and to promote that research results come to use. And one obvious way to do this is, of course, through commercialization, by generation of spin-out and companies based on research results. This has in Uppsala been especially successful within life science, but also in other fields of research. Further, in Sweden, the law regarding the rights to employees' inventions has a notable exception regarding universities, commonly referred to as the professor's privilege or the Swedish Teachers Exemption, effectively meaning that academic researchers own their research results, and the scientists retain the commercial rights to their research results. And this has shaped the support structure for scientists connected to the universities in a unique way. The university do not have tech transfer offices in a traditional sense, but they have functions that provide free of charge advice regarding immaterial rights at an early stage to scientists. The universities actively support researchers to explore possibilities for entrepreneurship. Since the researcher owns the rights, it's always their wish to explore commercialization that will be determining. If the researcher wants to collaborate with the university holding company may finance early ideas in return for spin-out equity. But researchers can select other investors or partners depending on the preference and regarding to what degree they want to be involved in the commercialization. This setup has created a huge incentive for Swedish researchers to drive the development of innovative ideas. And the system has proven to be effective. Sweden is once again top ranked within the European Union in regards to innovation. The European Innovation Scoreboard 2020 stated as Sweden continues to be the innovation leader, followed by Finland, Denmark, and the Netherlands. On the regional level, Uppsala has a dedicated support system for researchers who want to develop ideas and to find partners for their ambitions as entrepreneurs. Finally, Sweden, and in particular Uppsala, is a place of special interest to watch for finding early tech, research partners, and new exciting projects, both within life science and in other fields. Thank you.
Thank you. Thank you very much, Mats, for excellent presentation here. I hand over to Dr. Henrik Johnson.
Thanks, Ola. It's great to be in this panel, and I'm live from House of Sweden in Washington, D.C., together with the ambassador today. We have Snow here as well. So I'm very touched to see the bold effort to accelerate progress in cancer research. So thank you, everybody, for all the efforts and contribution you do. So is Sweden different in terms of a unique innovation model? I would like to, within a few minutes, just to take a more high-level approach to the Swedish model and look what US is doing in terms of that and see if we can improve. So based on the results, we obviously do something interesting up in the Nordics, and in general in Europe, 2/3 of the European growth is coming from research and innovation. So, and I think the Swedish society has long been fostering innovation and entrepreneurship. With this comes a curiosity where new ideas, trends, and technologies are welcome. And however, the Swedish market is simply too small and forced us early to go global, both in terms of trade and research collaboration. In Sweden, the big international companies dominate the R&D system. About 70% of the funding is from the companies. And the university, as you heard here, for example, Uppsala, dominates the public R&D. And they also have the third task to cooperate with the companies and the society. Sweden's long-term focus on education has also made an impact on the capacity for innovation. Already in 1842, Sweden introduced compulsory schooling. That was a game-changing move. And today we have over 400,000 students in higher education in Sweden. That's an impressive number with population of about 10 million people. Recently, just before Christmas, the Swedish government presented the new research bill, and we now reach about 3.4% of the Swedish GDP in research and development. That means we're number one in Europe. United States spends about 2.8%. That's also an impressive number if you look on the dollars. They spend about $550 billion on R&D, 400 from the industry and about $150 billion from federal. Sweden, we also have a very known welfare state that is also beneficial. It offers the freedom for entrepreneurs to experiment and take risks. And we have a high degree of equality. We still improve and can do better. And we have an infrastructure already from the 90s. The Swedish government pushed out widely developed broadband network, and we also subsidize computer lending programs, etc., to work on the digital divide. The digital divide is in the United States a very big issue, and I think we will see new policies coming out based on that issue. We have the large-scale infrastructure in Sweden, ESS, Max4, SciLife Lab, etc. Now quickly look on the US side. There are a number of key elements of success. Strategic focus. Look at the President Biden's new science team that was launched recently, led by Dr. Eric Lander. And President Biden wrote him a letter. President Roosevelt did the same with 5 to 6 questions at that time to his science advisor, which was Dr. Bush. That created the foundation to the NSF and all the innovations we've seen in the United States today. So I highly recommend, look at the 5 questions President Biden sent to Dr. Lander recently. They have commitment for long-term and sufficient public and private funding. Just quickly look at the importance of large-scale federal research funding in the US after World War II. That has historically created disruptive technologies as the internet, the GPS, etc. In addition, federal funding is then the very foundation to the many successful US innovation companies that we have today. 13 out of the 16 largest tech companies in the world are Americans. You know them, Apple, Google, Amazon, etc. The US also managed to work and create success stories based on dual use, which means that the military research became a tool also for civil applications. Strongly recommend you to look at the DARPA initiatives and how they accept to fail. They're also able to attract talent. I think mobility is maybe the most important issue that we have to deal with today. We need to increase mobility between continents and countries. Look at the Nobel laureates America managed to attract and support. 40% of the laureates since 1901 are Americans or have an American affiliation to an institute. Last year, they had 8 American laureates. This year, another 8. It's very impressive to see. The United States has also created 17 national labs run by the Department of Energy. And we are in the middle of a process now to start and increase our collaboration with the Swedish infrastructure mentioned before and the United States. They also connected around that this surrounding ecosystem, and we have similar now in Lund around ESS with the Science Village Scandinavia. The White House recently also announced a $1 billion investment in quantum and AI, and they're building up 12 research institutes highly connected to the national labs. Finally, it's hard to have excellent research and innovation being excellent without excellent education. Silicon Valley is a beautiful example of this in the United States, in which the university played a central role. There are several other interesting regions as well. The United States has about 3,000 colleges and universities. So my key message, or today's tweet, we take international collaboration for granted. It happens anyway. By that, we do not scale. We need to continue to stimulate collaboration and mobility, bring the future leaders out in the world to build bridges. So I stop here, and I will be happy to, maybe not in this event, in the future event, to also talk more about why is US universities so successful, what are my observations, etc., etc. So thank you very much. I hand it over, Ola.
Thank you very much, Henrik. Excellent. Excellent job, all the three of you. So now we have set aside some minutes here to have a little discussion. I have a couple of questions. So I will actually start asking Mats. We started talking about this teachers exemption rule. That clearly seems to be very attractive for individual researchers, but what's in it for the Swedish universities and how could this keep on going?
I mean, the government expects the universities to produce innovations which will give new companies and commercial successes like Oncopeptides. So that's the basic reason why the government is supporting this system. I think it's a great system.
So wouldn't the Swedish universities benefit if all these patents from new innovations were owned by the universities? Why is that not the principle?
I think one difference is that universities in Sweden are public. They're owned by the government. So the government finances us to a large extent, and they expect us to produce innovations that are beneficial for society. So that's the background, that's the Swedish model.
So I'm going to turn to Henrik and say, with this very active life science sector in Sweden, what is your perspective from the US, coming from Sweden, representing Sweden, or American colleagues, collaborators, various parts of the American system aware of these ways of working from the Swedish Life Science program?
I think they are aware, but they're also very comfortable, and I think they appreciate the support they get from the US universities around. The ecosystem here brings in a lot of funding. If you look at the Boston area, the Silicon Valley, you have a strong combination of private funding and federal funding together with the university's support. So what I've heard so far is that they're also very happy with the system over here, but we have a unique system in Sweden that we should be very proud of. But of course, as in all systems, I think we can develop it further.
So Henrik, are there any other research or industrial sectors that are important for the Swedish embassy in the US?
Yes, there are a number of key areas, especially now with the former administration. They focused a lot on emerging technologies, AI, quantum, biotech, etc. And we see that now, of course, also with the Biden administration. Maybe we will have even more international collaboration. And the life science sector is one of them. And we already have one agreement with the Cancer Moonshot program, as the ambassador mentioned. And I think the cancer area, as we talk about today, is a key area, and it's also a key area for for Europe. It's one of the mission areas in the European Commissions.
Thank you. So maybe I turn to Joachim. So Joachim, there seems clearly to be the incentive for the individual academic researcher in Sweden to start a company, and you are the prime example of this. I think looking at some of the statistics for a number of patients on clinical studies over the past 20 years or so, that those numbers have been going on. How could that be? Why is that not just more and more? Do you have any thoughts on that?
Yes, as there are more and more standard options to treat patients and a lot of competition in the area, and also development outside clinical trials and empirical observation, I think it's a very tough competition in the drug development area because there are so many options, and that's good for the patients in the end.
So what role do you see that the Swedish life science has to play here in the US in the future?
I hope that this model would benefit more projects and more collaborations, and I think we could benefit from academic collaborations as well. You mentioned the incentive to do research this way, and I think for most academic researchers, the incentive is not commercial or about money. It's academic curiosity. You just want to know how and why things happen. And then there's a bridge. Can this be applied? And then the company perspective is, of course, how can we make a profit out of this? And I hope that a lot of ideas travels this bridge.
Maybe I ask the same question to Henrik and Mats. I'll start with Henrik. So where do you see the Swedish life science could play a role here in the US in the future?
I think in terms of what we see now in the pandemic, we really need to continue collaborate, and that's in all sectors. This is an interdisciplinary problem to solve a pandemic. Cancer is an interdisciplinary problem. They have an amazing experience over here working with the big national labs. I'm really looking forward to see what Sweden can get out from our national labs on SciLife Lab, ESS, Max4. So I'm really eager to find out more about what we can do in terms of collaboration. I'm very happy with the new administration's approach to increase collaboration globally to solve the big challenges we have in front of us.
Thank you. Mats, what do you think the Swedish life science, the role it has to play in the US in the future?
Well, if I turn the question around a little bit, Uppsala University is a large university in Sweden, but the world is big and we need to collaborate. So for us, it's very, very important to have research collaborations with American universities. I also think that innovations that are created, for example, in Sweden, again, Sweden is a small country, and really to develop, for example, innovations in the pharmaceutical area, you need to think globally, and then you need, of course, to have collaborations with US.
Thank you so much. I would like to thank all the three of you for outstanding introductions here, and thank you so much for participating in the discussion. Thank you so much. And with that, I would like to wrap up this first panel, and I would like to move on to the next section, which will be a short little movie here.
So we had a team that was convinced, but our ownership base was split. And one of our biggest owners, they were very skeptical about the molecule, and they were cutting the funds.
So we were in that difficult situation where industry funding was saying yes, and Karolinska Institute, Karolinska Developments were saying maybe, but we're not convinced.
It's very rarely the science that tips a company over. It is all the other things around it.
How do we get rid of these owners? Because you cannot just get rid of your owners. You actually need their signature on a piece of paper to make that happen. So we were facing a steep challenge at the time. In the end, it boiled down to emotional arguments. Please, don't let us die. Eventually, these emotional arguments actually worked, and they sold their shares. We financed the company to do the clinical development program the day after.
We were persistent. We carried on. Even the hospitals actually charged us less than what they do to big pharma, because they also believed in the molecule.
In record time, we got listed on the main stock exchange here in Stockholm. It was a crazy amount of work. We entered the main market. We were in the same segment as large household names on the stock market. And we were 15 happy-go-lucky people in a room.
It's just amazing, I think. A Swedish molecule having been taken this far in development.
It's the passion, it's the courage, it's the collaboration, it's the trust, and it all starts with the science. We're calling it One Oncopeptides.
This is what you're trained to do. This is why you spend so many years in university.
I mean, it's great that we have developed a new drug, but there's so much more to do. So I guess you need to be very humble. The patient relies on people such as us to try to push the science, but at the same time not promise them sort of eternal life and happy sunshine, because they are in a very, very tough spot.
What I've learned is that you can do a lot if you put your mind to it. It's been hard at times, and you have to ask the right people for help, but it's amazing when you actually see the results of all the efforts that you put in.
I mean, the whole reason why I am in this industry is because we're trying hard to provide people with time, that is moments. and we try to give it to them as much as we can. And I think that's a very worthy endeavor. If you want to understand what Joachim Gullbo added in the lab, you can look no further than my lower left arm, because I have the molecule here. To make it simple, this is the alkylating portion of the molecule. That is the old drug, 70 years old. This is the portion that Joachim Gullbo added, and that is the portion that makes all the difference.
Thank you so much, and welcome back. We will now move on to the second of the two planned panel discussions. Focus for this panel will be a Swedish-US research collaboration driving innovation in cancer. The goal of this discussion is to focus on the value of research collaborations and to address the roles of industry, academia, and society working together. We have 5 invited participants for this panel. Each of them will speak for 4 minutes, and then we will have time for about a 15-minute discussion. I would like to welcome Marty Duval from Oncopeptides, Professor Kenneth Andersson from Dana-Farber Cancer Institute, Jenny Nordborg, Swedish Ministry for Industry, Susanne Håkansson from AstraZeneca, and Professor Tobias Sjöblom from Uppsala University. Warm welcome, everybody.
Thank you.
I would like to give each of the speakers about 4 minutes to give a brief background, and I will start with Marty. Marty, the floor is yours.
Thanks, Ola. And with the weather, I was trying to get to Washington, D.C. to be in Sweden, and here I sit about 30 miles north of you in Miami. So I'm in Coconut Creek, Florida. But anyway, I'm glad to be here. And on behalf of Oncopeptides, it's my pleasure to participate in today's program. You know, our company brings hope to patients through passionate people, innovative science, and transformative medicines. And at the core of it all is the innovative science driven by Joachim Gullbo and the academic collaboration first initiated in Sweden between the Uppsala University and the Karolinska. But that program really began to take shape through the Sweden-US research collaboration with the Dana-Farber Cancer Institute in Boston. And that continues to be a key driver of our innovation today. Preclinical and clinical work at Dana-Farber truly complemented the efforts in Stockholm and has blossomed today into an extensive global research collaboration. So this extensive research collaboration now encompasses multiple phase 3 trials and a wide and growing development plan for our lead drug. The research network currently includes 34 US clinical sites and 131 European clinical sites, including 9 in Scandinavia. And as you can see in the bottom left hand of the graphic here, there are a wide range of research collaborations with 15 in the US and 20 plus across Europe. Now, these research collaborations are also driving scientific publications. So from 2017 to 2020, there were approximately 85 scientific publications on preclinical and clinical data highlighting the Oncopeptides peptide drug conjugate platform. From the graph on the bottom, you see a sharp growth in publications and our ambition of reaching a new high of nearly 70 publications in 2021 alone. And this work lays foundation for funding and expansion. This was most recently realized in the spring of 2020, when on the heels of strong clinical data in relapse refractory multiple myeloma, Oncopeptides raised about $150 million to fuel the development of our pipeline. In turn, this creates jobs and career opportunities for researchers, scientists, and business partners in both Sweden and the US to advance the mission to impact patients affected by cancer. We grew in 2020 30% each quarter, so on a quarter-by-quarter basis, and we currently sit at 280 employees, now split equally between Sweden and the US. And mirroring the locations in the heart of our research endeavors, our company's headquarters in Stockholm and Boston, respectively. And we soon plan to build out more across Europe. And finally, I'm excited to announce that we're in the early stages of creating a scholarship program. This program will be aimed at continuing to promote the Swedish-US research collaboration in cancer research with the importance of the Uppsala University and the Dana-Farber Cancer Institute as a model. So we'll have more information on this as we move into the spring. So that's it for me, and Ola, back to you.
Thank you so much, Marty. Exciting, very, very exciting. I hand back to Boston. Ken, may the force be with you.
Thanks so much, Ola. I've worked on bench to bedside research leading to new treatments for myeloma for over 4 decades now at Dana-Farber and Harvard Medical School. And I'm particularly honored to be here today to affirm how this Swedish innovation model of close collaboration between industry, research institutions, and society in Sweden can drive innovation and move scientific advances from the bench to the bedside to benefit patients internationally. I want to start by saying that it's a particular honor for me, as I'm of Swedish heritage. Both my paternal and maternal grandparents emigrated from Sweden to the United States. In my earlier academic career, I focused on blood banking at our cancer center, and I was invited to speak in 1989 at the plenary session of the European Society of Hemophilia in Uppsala. While at the university there, I sought out Kenneth Nielsen, an eminent professor of pathology who derived the U266 cell line from a Swedish myeloma patient, a myeloma cell line used by me and investigators around the world. Kenneth was delightful, and I learned that we not only shared a common interest in myeloma, but we also had common family roots in Sweden. His wife's and my mother's families trace back to the early 1700s, not only from the same church parish, but also from the same farmhouse in Värmland. As you know, melflufen grew out of the discovery and innovative research of Joachim Gulbo at Uppsala in the late 1990s. At that time, I, with my laboratory colleagues, Teru Hideshima and Dharminder Chauhan, as well as my clinical colleague, Paul Richardson, carried out preclinical and clinical trials leading to the regulatory approval of the proteasome inhibitor bortezomib to treat myeloma. As you also know, the Nobel Prize in Chemistry was awarded in 2004 for the discovery of the proteasome. Because of our translational studies targeting protein homeostasis, I was invited to speak at a New Frontiers in Cancer Research and Therapy Nobel Forum at Karolinska in 2009. And it's there that I met Rolf Lewensen and Christina Victorsen from the Department of Oncology Pathology at Karolinska and first learned about melflufen. As we discussed this exciting discovery and development, we decided to collaborate and evaluate melflufen in our preclinical and animal models of myeloma, which in turn would provide the scientific basis for clinical trials evaluating melflufen to treat myeloma patients. I'm proud to say that this has led to a journey of over a decade of collaborative preclinical laboratory and animal studies with multiple joint publications demonstrating its efficacy and tolerability. And most importantly, these studies have in turn derived multiple international trials spearheaded by Paul Richardson, resulting in the imminent regulatory approval of melflufen to treat patients with myeloma. This highly innovative Swedish innovation model is quite literally a paradigm of how to make scientific discoveries count for patients and assures that promising approaches discovered in academia are developed and clinically and tested to improve medical care. So many times in the past, promising discoveries have never transitioned to clinical evaluation due to the risks and associated costs. Moreover, new drug development requires teamwork, academic institutions, biotech and industry, regulatory authorities, funding agencies, and most importantly, patients to drive discovery and innovation from the bench to the bedside. Importantly, this Swedish-US collaboration represented here is a testament to the value of international collaborations to even further fast-forward progress and has led to a novel medicine that will help myeloma patients and their families worldwide. So, especially in the midst of a worldwide COVID-19 the importance of such international collaborations to assure the rapid translation of scientific advances and access to patients everywhere has never been greater. Our Swedish-American academic Oncopeptides collaboration has filled an unmet medical need and given the gift of hope and of life to myeloma patients and their families worldwide. There is no greater gift than this. Finally, I've never been prouder of my Swedish heritage, and I want to extend my heartfelt congratulations and thanks to all on the team that have made this wonderful advance possible.
Thank you very much, Ken. Very well said. Thank you so much, fellow Swedish myeloma researcher physician. I will now hand over to Jenny Nordborg from the Swedish Ministry for Industry.
Thank you so much. It's a great pleasure to be able to attend this seminar today. Thank you so much, Oncopeptides and the Embassy in Washington, for organizing. It has been a true pleasure for many years to be able to follow the development of Oncopeptides as a platform company and this field as a whole, and to see the all fantastic breakthroughs that are now on the verge that makes hope for the future. And especially happy to be able also to talk about the Sweden-US collaboration and what we could also foresee for the future. And I would like to pick up on the presentation also from the previous panel and Henrik Johnson, who really stressed that if we don't focus on collaboration, we won't scale. So we also need to see how can we really utilize the fantastic collaboration that you have between you as an infrastructure also to scale within other areas, both from Sweden and from the US. So my mission as a national coordinator for life sciences is really to have a dialogue, both nationally and internationally, within the field of life sciences broadly, and with a focus on precision medicine and health data. I work for both the Minister of Research and Education, Minister of Social Affairs, and Minister of Business and Innovation, with the long-term national strategy as a platform for collaboration. I do think that the long-standing tradition that we have between Sweden and the US, with the cancer moonshot as an example, but also the driven action for innovation between our two countries are really essential also in moving forward. So what does Sweden really bring to the table? Well, of course, the excellence in science, but also I think that we can build even further on the excellence in collaboration and in implementation, being both, having the ability to be both adaptive and agile when it comes to new research and new ways of collaboration. Sweden opens up for great opportunities to utilize real-world data, moving towards real-world evidence. And I think now when we are at the verge of really seeing the explosion of precision medicine moving towards a even more data-driven life science industry, I do think that we have a lot to offer. Also with the strong startup community, platform companies like Oncopeptides who really go global from day one. And hopefully Sweden can offer a hub for innovation and data-driven life sciences where we can work in close collaboration with implementation also of precision diagnostics, really utilizing the new opportunities that we see with artificial intelligence and implementation in life sciences. So to conclude, it has never been more evident than during the pandemic that we need new types of partnerships. We need stronger partnerships, reinforced partnerships, really driving innovation to tackle the big societal issues when it comes to infectious disease, but also when it comes to tackle the unmet medical needs within cancer. And I was so happy to hear also the announcement that you just made on Oncopeptides with the new scholarships that can also reinforce our collaboration for the future, being able to attract more talents to this field as a whole. With that, thank you.
Thank you very much. Thank you very much for another excellent presentation. We have 2 more to go before the panel discussion. So I would like to invite Susanne Håkansson from AstraZeneca.
Yes, thank you so much for having me. What a great seminar. And also fantastic to hear about Oncopeptides and your story and your development. AstraZeneca is a more mature and a bit older company, but also ever changing. We are a global biopharmaceutical company focusing on 3 therapeutic areas: cardiovascular and metabolic disease, respiratory, and oncology. But now also, as the ambassador also mentioned, we're into vaccines and infectious disease. We have global presence and collaborations, but our 3 major, the biggest science hubs are located in the US, in Gaithersburg, in the UK, in Cambridge, and in Sweden, in Gothenburg. We are science-led, and we are truly innovation and patient-centricity oriented, and therefore collaboration and co-creation are key in all activities throughout the company. In oncology, our research is focused on 4 scientific platforms targeting the underlying biological drivers of cancer. We do have a precision medicine approach concentrating on biomarker-driven indications, and we strive to dramatically improve 5-year survival in especially 5 tumor types, including ovarian cancer and non-small cell lung cancer. And as I said, we're patient-centric, and this means mainly 2 things. And the first thing is that we try to identify and help treating patients as early as possible, which really takes a lot of caution, of course, with healthcare and with others. And also to support relapsed and refractory patients by tackling resistance later on in the treatment journey. Science is, of course, collaborative by its nature, but also all over the world right now, we are trying to work more from an ecosystem perspective and in a new way, actually, coming closer to the patient, collaborating with more different kind of actors and in partnerships. And this is to try to harness the potential that lies in new medical and diagnostic solutions and other technology and data progress that we see. And I want to share 2 examples of collaborations with you today, which are ecosystem-oriented. And they are both within the field of lung cancer. Lung cancer, as you know, is the leading cause of cancer-related deaths. And actually, every 18 seconds, a person dies from lung cancer right now. And still today, as many as around 40% of patients are detected only late in their disease journey. They are diagnosed after the disease has spread beyond the lungs. So therefore, we have started collaborating in new ways. And one example is our global partnership, Lung Ambition Alliance, which was shaped to change the pace in lung cancer survival. The long-term objective is actually to eliminate lung cancer as a cause of death and to double 5-year survival in the shorter term to 2025, actually. Today, only 1 out of 5 patients die after 5 years after they are diagnosed. We do this together with partners, with a diagnostic company, with a global patient organization, the International Association for the Study of Lung Cancer. And now more pharmaceutical companies are also joining this initiative. In Sweden, we have a similar, and actually it was started before the Lung Ambition, we have a similar initiative called the Vision Zero Cancer Initiative, where industry, healthcare, academia, and patient organizations have joined forces since the fall of 2019. We are working with the joint vision to actually also to transform cancer to a curable or a chronic disease. And this is truly according to the triple helix model that has been mentioned here today a couple of times. The initiative is co-funded by the government through the innovation agency of Vinnova. And what we do is that we jointly address the whole patient pathway from prevention and early detection and diagnosis to high quality treatment and follow up, to economics, organization and leadership of oncology care. And we do together, and we try to spread the labor between the organizations and do what we're best at and collaborate the best we can. Vision Zero Cancer, of course, like AstraZeneca, wants to learn from the best environments in the world. So we're really, really happy to hear more about any collaboration alternatives and possibilities. And we want to collaborate with the most ambitious partners and the innovative approaches we can find. So looking forward to the discussion coming up. Thank you.
Thank you very much. I hand over now to Tobias Sjöblom from Uppsala University.
Thank you very much. So thanks for having me. I'll now proceed to wear no less than 3 hats in less than 4 minutes. Quite a feat in and of itself. So the first one is relating to the slide, research excellence. So as a cancer researcher myself and as head of department, I'm happy to represent the largest recipient department of grants from the Swedish Cancer Foundation. So Sweden has several strong cancer research institutions that you've heard about today, such as the Karolinska Institute, Uppsala University, and also Lund University, but would benefit from collaborating more, particularly with the US. A second hat, samples and data. This is a Swedish strong point. So as director of Biobank Sweden, I can testify that our country has great infrastructure for translational research in cancer. At the national level, we, the universities, join forces with the healthcare providers in Biobank Sweden, which is an infrastructure to streamline and accelerate biobank services to users, including the build-up of a national sample registry linking all samples in healthcare to enable their linking to clinical data. And a more specific example is the UCAN Biobank here at Uppsala University, which holds more than 20,000 patients or more than 10 different tumor types included since 2010. UCAN has so far provided materials and data to more than 100 studies in academia, pharma, and diagnostics industries, including in the US. And this is just one example of several large Swedish cohorts available for collaborative cancer research. And now for clinical data, we have a great basic infrastructure, but more recently, together with the healthcare providers, we have fielded a nationwide real-time registry or decision support system for oncology, which provides structured high-resolution data, treatment data from these often protracted care processes in oncology. And this is a unique effort that will serve to the benefit of patients, healthcare providers, academia, and corporate R&D. Now, all these samples and data can be drivers for collaborative research across the Atlantic. I and many with me think that we need more financial and intellectual firepower to realize their full value. Now, my final hat would be as an entrepreneur. So having myself started several companies based on our research, not just as successful as Joachim Gullbo's endeavor here, Oncopeptides, I can assure you that the Stockholm-Uppsala region is one of the largest life science clusters in Europe, with more than 600 companies and 20,000 employees. For example, our business incubator, Uppsala Innovation Center, is ranked top 5 globally among university-affiliated incubators by value creation in the incubated companies. I think we have a great fundament here. And I think also that Oncopeptides is a great example of successful commercialization of academic research from our region. However, also in this area, we will benefit from even more venture capital in the life science area. So with that, I will now stack my 3 hats on top of each other and look forward to the panel discussion. Thank you.
Thank you very much. Thank you very much, all of you, for great presentations. So now we turn into the next part here of the panel, which is the open question back and forth. So I will start asking Jenny Nordborg. So I, in preparation for this seminar, read up on some of the literature, and there's a recent pipeline report from Sweden Bio. I believe in 2017, Sweden was on third place in the world after China and the US in terms of new life science listings. And I think, according to this report, around 15 or 13% were coming from the Stockholm Nasdaq stock exchange. And there's also been an increase by 7-8 new listed life science companies every year. So that's very impressive. So do you have any information on how many of these that originate back from academic inventions? Do we have any such benchmark anywhere? Do you know?
I must confess I don't know the data on that, but I think that within life sciences it's so much higher than in any other field, and a lot of the companies, at least from the sort of basic life sciences companies and the ones that you've listed, and those that are enrolled in that pipeline study, they definitely have a science origin, and in that sense, these are both good and bad figures, as you know. I mean, early listings and the high quantity of listings on the Stockholm Nasdaq is, of course, due to, as Tobias Sjöholm also stressed, for scaling companies. The Swedish area of field for venture capital is maybe does not really apply for that sort of scaling that is needed.
Yeah, that's very, 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 very true. So is there any data on the success rate? How many inventions reach the market? Do we know that?
I'm not aware of the certain data, but I think that the sort of old school figure of one of 10 still applies in that sense, and I fear that it might also be when it goes to very early listings, if you count on that, it would become even lower. But it's more of, I think, of interest. I mean, what is success? And if you're looking at success when it comes to scaling, of course, we see too few of that, but we really need to focus on those few that actually coming to scale.
Right. Yeah, those are very important aspects. I will move on and ask Marty Duval. So you are in the mega life science hub in the US up in Boston, and you have experience both from big pharma and small biotech. Are there any similarities or differences that you see if you compare US and Swedish life science strategies and how these companies operate? Could you shed some light on that?
Sure. Yeah. And thank you for the question. And I would say mostly I see similarities. And I think the common point is as Jacob Lindberg pointed out in the video, if it's good for the patient, it's good for Oncopeptides. I truly see the commonality around the world, the focus on patients, advancing science and improving patient care being key. I am learning a little bit more about some of the differences on the financing side and the governance side. We are a company that's listed NASDAQ Stockholm. And there are some unique differences, but also some similarities regarding, governance of the companies but I think the appetite to develop the technology, move the technology forward, the access to capital seems to be very good, actually in Stockholm, as it is, of course, as a US-based or NASDAQ US-listed companies. So I think those are all the good aspects. We're certainly looking at the same pool of patients and certainly the excellent academic partners that we're all interested that we know can help advance our science.
So one question that came in through the chat here to me is, in this model with the teachers exception rule, could you shed light on the role of venture capital? Is it different or is it not different in the Swedish model?
Is that directed to me, Ola?
Yeah.
I think it's pretty similar. Yeah, pretty similar. I don't see a big difference in that particular from that particular frame.
Thank you, Marty. So I will move on and ask Susanne Håkansson. So you represent one of the largest pharmaceutical companies in the world, and you have large internal research programs going. So how is your organization dependent on international research collaboration and life sciences strategies in individual countries? Could you shed light on that?
Yes, I would say my response to that is that we are totally dependent on working with, groups of scientists and groups also within our own company across the world. We're a true global company, picking up talent and picking up interesting research, also collaborating with small and large firms within, of course, medical science, but also increasingly with different kind of other technologies, with diagnostics and health data companies and so on across the world. So highly, depending upon international collaboration internally and externally. And I think there's a lot of things. I think we're on a path of learning on how to collaborate in a really good way. It takes a lot of listening and understanding and a lot of trust, and also finding people with true drive to move processes and projects forward. And also similar, this last thing, we talk about venture capital and all the small companies here, but also for us, of course, a big challenge is to understand what projects to proceed with and which ones to actually exit, which is a really difficult thing for I think, all companies.
Thank you. So I have a question for Tobias Sjöblom. So Sweden, you mentioned, is unique in many ways from a research perspective and beyond. So you have access to a range of these governmental funded high quality nationwide population-based registries. That was one of the bullets you had on your slide. Are there other countries that are developing the same type of strategies, or is Sweden still unique when it comes to that?
No, I think, Ola, this is of course a lot of countries running after this, and we need to be one step ahead. And I think what we're doing in oncology now is truly unique, at least by European standards, that we have now actually fielded a nationwide information system to capture these therapy data, which has so far not really been efficiently incorporated in registries. So we have to move ahead. We can't just rely on our past victories. We need to press forward.
So sharing a little bit of my own experience being raised and trained initially in Sweden, coming here to the US, when I started looking into registries here in the US, I compared them with Swedish registries. No one really is kind of fleshing out all the details when they publish them, at least not to the degree that people fully understand how big differences I think there are. So Sweden captures every single person in the whole country. Here in the US, you could have very detailed registries, but they maybe reflect 5% of the population. So in my practical experience, I think a lot comes back to when you write up and describe these types of research projects and papers and things like that, to be very specific and outline how they are unique. That's something I felt very much people didn't fully understand how unique many of these resources are. So Tobias, can Sweden offer US collaborators things that you cannot find in other countries?
Well, it depends on who the collaborator is and what they want to do, but what we can and what we do provide, for example, in our collaborations, is access to vast amounts of information and samples in collaborative research, also for analysis in the US, for example. So all this is feasible, and I think, of course, depending on what you're going to do, you source your samples and data from different sources. But I can see that we attract a lot of interest now from other large entrants sort of on the scene, particularly China, that have sort of realized the potential of Swedish registries and samples. And you would imagine that China would have enough samples and registries, but it's clear that they don't. And but I mean, I think we have a long history of collaborating with the US, and with the steps forward in the US science policy, we look forward to working more closely with US companies and the US academics.
Thank you. So I turn next to my fellow Swedish myeloma collaborator in Boston, Ken. So Ken, we have so many cultural and linguistic and economical and other barriers in the world. So why is it important with these international research collaborations in the future?
Well, I think, to be honest, Ola, we owe it to the patients. I think that what everybody here in this whole WebEx has said this morning is if you put the patients first, no matter where you're working, it requires that we take basic discoveries, as Oncopeptides so beautifully represents, and make it count for patients, make science count for patients. You know, we all work at different universities, and some of us work in Sweden and then work in the crazy United States. But in any event, we all have goals, and we see barriers to those goals. In academia, we get promoted and get grants and get titles, and companies can be formed and be successful. But to be honest, the success that we all share is improving the treatment and outcome for patients and their families. And this is not limited to any one country. And as what we can testify, Ola, when you really start to focus, so let's focus on myeloma, and let's think about people that are trying to do new things, or companies, Oncopeptides, that are trying to do new things in myeloma, it ends up being a small number. And you end up having personal and professional collaborators. So I think the reason that there are many barriers is there's too much isolation, and too many times people are not focusing on the correct goal. The correct goal is to making advances, discovery to the patient, and number two, which we haven't talked that much about, making sure that there's access of these advances. So when melflufen gets approved imminently in the crazy United States, there has to be access not only in the US, but this can be an example, I hope, for subsequent approvals and access all around the world, because that's really success, right? Improving the health of patients forever. I think that has to be our very high but very reasonable goal.
Very well said, Ken. Yeah, I think you summarized it very well. It's all about persistence, working hard, focus, always focus on patient improved outcome, and helping each other to advance the field. We cannot do any of this work as individuals. We need each other. We need to work together and help each other to leverage to reach these goals. With that, I would like to thank all the participants for being part of this panel discussion. Thank you so much. And I'm going to move on to the last part of this program. So thank you. I would like to have a few minutes of concluding remarks, and then I will hand over to the Swedish Embassy in Washington, D.C. We're almost at the end of the program. We have about 5 minutes left. So I would like to thank you all very much for attending this Swedish-American journey of innovation in cancer research. It's been my pleasure to serve as the moderator for the program. As you have seen and heard through these lively panel discussions, and also many questions that have come in, this is clearly an area where there is more to discuss. I've received a lot of questions via Zoom, and we try to answer some of them during the panel discussions. But because of the great interest, we will have to follow up and answer remaining questions at a later time. So therefore, you will receive an email in the next 2 weeks where you will be given the opportunity to join a connection session if you're interested. I hope you have enjoyed this program. Ha en bra dag. Have a great day. Before we end, I would like to hand over to Washington, D.C. and the Ambassador of Sweden in the United States, Karin Olofsdotter, for her concluding remarks.
Well, thank you all so much for participating in today's seminar, and thank you, Ola, for moderating the discussions. And also thank you to our partners to this event, Dana-Farber, Harvard University, Uppsala University, and Oncopeptides. As we learned today, cancer affects everyone regardless of age, gender, or social status, and of course represents a tremendous burden for patients, families, and societies at large. And if no further action is taken, the number of people newly diagnosed with cancer every year in Europe will increase from the current 3.5 million to more than 4.3 million by 2035. So investment in research and innovation is, of course, then crucial towards achieving our goal to beat cancer, contributing to the development of effective healthcare and tools for the prevention, early diagnosis, and successful treatment of the disease. By joining efforts, we will ensure that more people will live without cancer in the future, while increasing quality of life for those affected. I also think our discussions really showed the importance and potential of further collaboration between Sweden and the US in the life science sector. And I'm sure the examples we have heard today will be of inspiration to others. And from the embassy side, we will continue to deepen and broaden the collaboration with the US in this area. I also want to mention that we will continue with webinars from House of Sweden on topics as culture, sustainability, science and innovation, trade, etc. So please do follow us on social media platforms and make sure to join us for upcoming events. Thank you so much for today and have a continued nice day. Thanks a lot.